HIV survivable. But not everyone has access to that treatment, and not everyone sort of wants to take it either. So about 25% of all the people around the world infected with HIV are not receiving antiretroviral therapy. And so those people are not likely to survive. There isn't anyone who's known to survive HIV infection unless you get a bone transplant. That's been in the news recently. 3 people have received bone marrow transplants to make them immune to HIV. So it's a really massive global health problem. There's many people dying every year. It is going down because fortunately we are getting more and more people on antiretroviral therapy, and you can use the therapy as what's called pre-exposure prophylaxis. You can take HIV drugs before you get infected and they can prevent you from getting infected, but that's hard and you have to take it all the time and it's expensive, and it's not really a global, it doesn't seem like it's necessarily a global solution, at least not to me. And all of that costs a lot of money. The United States government and many other governments pay a lot of money to fund antiretroviral therapy for lots of people around the world, which is great. But the more people that get infected, the more people need these drugs, and they have to take them every day for the rest of their life. So it's not clear that it's a sustainable solution to just be giving people drugs after they're infected or before they're infected. So, you know, this is the reason why we spend so much time trying to make a vaccine. It would work the way the COVID vaccines work, and it would actually have to work a little better because we have to prevent getting infected. But hopefully it would, you know, the idea would be to develop a vaccine that would protect against infection. And then you wouldn't have to take those. So HIV infects cells similarly in sort of a similar mechanism to the way COVID-19 does. On the surface of the virus, there are spike proteins that are totally analogous to the spike protein of the coronavirus. And what's different, I mean, there are lots of things that are different, but they affect different cells. Coronavirus infects cells that have an ACE2 receptor on its surface. HIV infects have a CD4 protein and a CCR5 protein on their surface. And HIV targets CD4 positive T cells, which are a critical part of your immune system. And so when it infects those cells and kills those cells, that's why it causes AIDS, acquired immune deficiency syndrome. And that's why people that get HIV, they don't usually die from the actual HIV infection. They die from all kinds of other infections because their immune system isn't working like it's supposed to. And so the vaccine needs to prevent the virus from infecting these host cells, just like the coronavirus vaccine is trying to prevent the coronavirus from infecting ACE2 cells like in your respiratory tract. So we want to induce, at least we spend a lot of time trying to induce neutralizing antibodies that will bind to the spike protein and prevent it from being able to attach to its cells and infect the human cells. And the tricky thing about HIV is that, so successful vaccines do induce neutralizing antibodies. Almost all the vaccines that you've taken, the major sort of effect of the vaccine is to induce neutralizing antibodies, including the COVID vaccine. They also induce T cell immunity, which is also important. But if you don't induce neutralizing antibodies, the vaccine is not likely to work. So we have to do that. For HIV, the big distinguishing factor, aside from the fact that it infects cells of the immune system and it's a retrovirus, meaning it integrates its genetic information into the genome of the cells it infects, which coronaviruses don't do, influenza doesn't do that. If you get infected with HIV, you can't get rid of it. It's in the DNA of the cells that have been infected. Aside from those two things, the major distinguishing factor is that the surface of the spike changes all the time. It changes when in one person, when they're infected, the daughter viruses have spikes that have a lot of differences on them. And from one person to the other that's infected, the spikes look very different. So HIV can mutate the spike much more rapidly than the of the coronavirus or of influenza. And that makes it very difficult to induce neutralizing antibodies that will, because and its neutralizing antibody might bind to one form of the spike, but then not to the next. And that's exemplified by the problem we've had with, Omicron. It isn't neutralized very well by the vaccines. It is neutralized still, but not as well as the earlier viruses. It's not neutralized as well as the earlier viruses by the antibodies that are induced by the original vaccine, which was based on the original virus, the Wuhan strain. And so for HIV, we have that problem of the spike diversity just manyfold. So this is a structural model of an HIV spike rotating around. It's a trimer, and the flickering blue structures are sugars, glycans, that are attached by the host cell when it's infected, and they looked kind of like self, so they help evade the immune system. And then in red, we've colored the positions on the spike that are just incredibly variable from one virus to the next. And so antibodies may very well bind to these. You could get a neutralizing antibody binding to these red regions, and that would neutralize that particular isolate, that particular virus, but it wouldn't neutralize the millions of other viruses, HIV viruses, that are circulating around the globe right now. So you have to induce neutralizing antibodies that don't touch the red stuff. And even the yellow region is difficult to check. There are changes there. So basically, the structure and the variability of the spike make it very difficult to make an HIV vaccine. And so this slide illustrates the sequence diversity, the difference in sequence diversity between SARS-CoV-2 spike sequences and HIV spike sequences. And so it's a two-dimensional graph, and both are on the same scale. So on the right, you've got it shows the genetic distance between different HIV spikes changed into different clades, A1, B, D, C. And these were just 1,000 different HIV isolates sampled in 2010. So if you were to take them all that are around the world right now, it'd be larger. And on the same scale, showing the same distance, the same difference in the spikes, number of mutations basically, and insertions and deletions, this little dot is the diversity of the SARS-CoV-2 spikes, 43,000 of them that were taken all around the world. And that when this was done, Omicron wasn't in there, but I guarantee that Omicron is still in that little dot. So you can imagine, if the challenge of making a vaccine is to make neutralizing antibodies that can handle all the viruses in this little dot, you can imagine how much more difficult it would be to induce neutralizing antibodies handling all these viruses. And my friends, Bethany Dearlove and Morgan Roland, made these graphs, and they also showed what happens if you zoom in on the SARS-CoV-2. You can still see there is a tree, there is a genetic relationship between the different spikes, and this dotted strain, that's the first one that came out. But you have to zoom in 75 times to see anything like this IV from 1 year. And it's also true, plotting on the same scale, we have trouble making an influenza vaccine. You have to take it every year and it doesn't work very well. We'd like to make a universal influenza vaccine that you only take, every once in a while, they can neutralize all the influenzas. And on the same scale, influenza is quite small compared to HIV. Same kind of thing. So that the diversity of the HIV really dwarfs the diversity of influenza, the spike protein, or SARS-CoV-2. However, people have been studying HIV for a really long time and trying to figure out, are there places on the surface of the spike that don't change very much? Places where if an antibody did bind, it wouldn't be the red parts that I showed you. If it did bind, maybe it could neutralize many different HIV viruses because that little patch wouldn't be changing very much. And many different people in the field, including several people at Scripps, have isolated what are called broadly neutralizing antibodies, or I'll call them BNAbs for short, and shown in this set of images as another model of a spike with different classes of broadly neutralizing antibodies bound to the spike, showing where they bind. So there's one binds right at the top. There's another set that bind to the high mannose patch, also sort of on the top, but not right at the apex. There are antibodies that bind in the CD4 binding site. So I mentioned that the spike, HIV spike, binds to CD4 receptors on T cells, and that's how it starts the process of infecting its target cells. Well, these antibodies bind to that same site where the CD4 protein would bind. So if this antibody is bound, the spike cannot bind to its receptor. There's just no, the binding patch is blocked. And I'll talk about this antibody more later. And there are other sites. So there are broadly neutralizing antibodies that do neutralize diverse isolates, some up to 99%. I'm just trying to move this over. Just going to summarize a lot of what we know. People in the field have done experiments with non-human primates where they would just administer a broadly neutralizing antibody directly, just give a monkey a shot and actually keep giving it shots to keep a sustained level in the blood, and then challenge with an infectious HIV variant that's been designed to infect monkeys called a SHIV. And BNAbs can provide sterilizing immunity. If you have enough BNAb, enough of these broadly neutralizing antibodies, high enough concentration in the blood, if you then expose an animal to an infection, it will not get infected. And we've done, this is a little bit of lingo from the field, but there have been clinical trials recently called the ANT trials in humans that did the same thing. They gave humans that participated in the clinical trial shots of this antibody VRC01, and they kept giving it over time to maintain a pretty good concentration in the blood. And they could show that not all of the people were protected, but it showed that some of them were, and they were protected if they were exposed to viruses that VRC01 could neutralize really well. So we know that we have proof of principle that if we could make an HIV vaccine that elicited broadly neutralizing antibodies, it could in principle prevent HIV infection. And to achieve that, the vaccine responses would have to be very broad. They'd have to be able to neutralize all these different isolates. They'd have to be very potent. They'd have to have a high concentration in the blood. And they have to be durable. They have to stay over time. And those are very difficult to achieve, but that is what the goal is. So our goal is to develop a vaccine that elicits sustained protective levels of broadly neutralizing antibodies. And we work particularly on the antibody part, but I mentioned T cells, and maybe you've heard of T cells related to the COVID vaccine. Good vaccines also induce T cells. What we want to get from the T cells is, if you think of antibodies are trying to prevent the virus from infecting any cells at all, and that may not be a perfect shield. There may be a few virus particles that make it through that shield and infect a few cells. T cells are designed to, they go around your body and they surveil the outside of a cell, and they can determine if a cell has been infected with a virus or not. Usually they can determine that. And for CD8 T cells, if they detect that the cell, your own cell, is infected with a virus, they drill holes in the cell and kill the cell to get rid of the virus. And so if we can elicit broad neutralizing antibodies and broad coverage T cells to sort of mop up anything that might squeak through the antibody shield, the field generally thinks that is what an HIV vaccine will look like. And that's what we're all collectively trying to achieve. It's so hard that everyone works on one part or the other. We're working on the broadly neutralizing antibody part. So, and even on the broadly neutralizing antibody part, we think that we need to elicit multiple different kinds of broadly neutralizing antibodies that bind to different sites on the spike, like I showed you before, in order to provide adequate coverage against the huge diversity of global isolates. So it's really tricky. It's tricky to elicit one kind, and we think we need to elicit multiple kinds. So how, what's going to be required? So at the end of the day, what we want the vaccine to do is to induce what are called plasma cells, and they end up in your bone marrow, and they will be secreting broadly neutralizing antibodies, and that will make broadly neutralizing antibodies in your blood all the time and in your organs. And so if you get exposed to HIV, these BNAbs would prevent infection in theory. But to get to that, how do we get to that? What vaccines usually interact with when you first receive them is they usually interact with what are called naive B cells. And you generate millions of new naive B cells every day, all of us do. And on every naive B cell, there's a different antibody on its surface. And only a very small fraction of the naive B cells that you have or that I have, only a very small fraction of them actually have the right genes that give them the potential to develop into a cell that would secrete BNAbs. Because you produce naive B cells that have different antibodies on their surface. Every B cell has one antibody, many copies of that one antibody. And each comes from different germline genes that are randomly joined together when that B cell is created. And you make hundreds of millions of different naive B cells because you get exposed to lots of different pathogens with lots of different structures on their surface. And our immune system has to be capable of binding to nearly anything. So you have the capacity, you have hundreds of millions of different naive B cells, and they are just waiting around to see if they run into some foreign antigen and so we need the vaccine to find where naive B cells that actually have the right genes that could turn into a BNAb. And we need to trigger them and get them to go through of immune reactions in which they will gain mutation, gain affinity so they can bind more tightly to the spike, and then convert into plasma. It's a very complex process. This is what vaccines always do. And this is what, when you get infected with a virus, this is what happens also. This process does occur. So it's a totally natural process. The really difficult thing with HIV is the place where you have to begin. Then there's very, very few naive B cells that are reasonable starting points. The vast majority of them, if your vaccine triggers them, this pathway won't to BNAb. And so we have to start from very particular, rare naive B cells, and they're very difficult to activate with HIV proteins. So how are we trying to do that? So there are multiple different strategies that people are pursuing, and I don't want to go through all, it'll be too complicated to go through all the different strategies. I just talked to you about our strategy, which is complicated all by itself. And we did test the first step in the human clinical trial that I'll tell you about. So I mentioned that we have to find the right naive B cells that have the right genes that give them the potential to develop into a broad neutralizing antibody. And for that, we design what's called the germline targeting priming immunogen. And its job is exactly that, to find those rare B cells and turn them on, generate a pool of memory and germinal center B cells, undergo one series of immune reactions. And then we need to come in with another immunogen. As part of the vaccine, it'd be like shot number 2, but it would actually be a different immunogen. And the job of that, we call that a vaccine boost 1 or shepherding immunogen. It's supposed to activate the memory B cells that were produced by the first vaccination, and it's supposed to trigger them and get them to undergo another series of immune reactions and mutate further, gaining more mutation and generate a new pool of memory B cells. And we think we actually probably need to do that several times with several different shepherding immunogens to teach the B cells to get the right mutations that will turn them into a broadly neutralizing antibody. And finally, we need what we call the last boost or a polishing immunogen to, once we've got basically broadly neutralizing antibodies in memory B cells, we need to convert them into plasma cells. And that's the job of this. All of these immunogens will be doing that a little bit, but the last one really has to do that because at this stage, you've got BNAbs and you need to get them in cells where they'll be secreted. So for the rest of the talk, I'm really just going to be talking about this first part because it's so difficult and this is a very new strategy. And it's very different from other vaccines that you take or that are licensed or that humans are used to taking or that pharmaceutical companies are used to administering. And so we had to find out, would this first step even work? In some ways, the first step is the hardest step. And so I'm going to skip this just to save time. So I mentioned this first step. This first step is to come with a germline targeting prime, find the right cells and generate a pool of memory B cells that are a little bit mutated in the right direction. And this step has to work in everybody you vaccinate. So consistent priming across vaccine recipients is a requirement. That's what I mean by that. Because if it doesn't, then that whole fancy sequence is not going to work, obviously. If you can't do the first step, the rest of the steps are dead in the water. And what that means is that for everyone watching this talk right now, you all have naive B cells because you're generating millions and millions of them every day, and they each have one antibody on their surface. The body has multiple different genes. It has a heavy chain and a light chain, and the heavy chain varies of 3 different genes. They're randomly selected from the different variable genes you have. And the light chain has 2 different variable genes, and they're randomly selected. So you make random different antibodies all the time. And then they have additional changes to make them even more diverse. So we all have the ability to sort of generate the entire human B cell repertoire. But at any one time, we're just generating one small subset of it. So in a vaccine, if you deliver a vaccine to 100 people, they don't all have the same naive B cells at that moment. They have lots of different ones. So your vaccine has to be able to find in each individual, they have a different set of naive B cells, and some fraction of their naive B cells will have the that could turn into a BNAb. This vaccine prime has to find those and turn them on. But the ones that they turn on in one person will be different than the ones they turn on in another person, because they will have some different. So it is very difficult. The priming immunogen needs appreciable affinity and avidity for diverse precursors, all that share some minimal set of properties that give them the ability to turn into BNAbs. And so we've been working on this for many different types of BNAbs, but for the longest, we've been working on these BNAbs called the VRC01 class BNAbs. These are the ones that bind to the CD4 binding site that I told you that block the ability of the spike to bind to CD4 T cells. And in order to bind that way, into the GP120 CD4 binding site, CD4BS, in order to do that, the heavy chain, so antibodies have a heavy chain and a light chain. I'm not sure how much you guys know about antibodies and genetics, and I'll just try to explain the minimal stuff that you need. So in order to bind this way into this CD4 binding site on this trimer, the heavy chain to use one of its genes is a VH12 gene. That's one of your heavy chain genes. It's the only way it can do it, and that particular gene has the ability to actually mimic the way that CD4 binds. It just happens, or it may be that we've evolved that way, but either way, that heavy chain gene is critical. So that's one property that precursors need to have. And also to squeeze in, it's actually a fairly narrow cavity on the trimer, to squeeze in there without bumping into the rest of the trimer. The light chain have, so light chains have a V gene and a J gene. And the place where they're joined together is called the CDRL3, the complementarity determining region on the light chain. And in order for this antibody to bind in this pocket, the CDRL3 has to be very short, only 5 amino acids long. The vast majority of your antibodies have CDRL3s that are 9 or 10 amino acids. So 5 is quite unusual. Only about 0.5% of human antibodies have 5 amino acids. So, but we're trying to find, our vaccine needs to trigger naive B cells that have these 2 genetic features. And we believe that a substantial fraction of all naive B cells that have these 2 genetic features do have the potential to develop eventually into broadly neutralizing antibodies. So our goal is to turn these on. And otherwise they have a lot of diverse other aspects of their sequence. So they have heavy chain CDR3s and different light chains. So we need a priming immunogen with appreciable affinity and avidity for diverse VRC01 class human naive precursors. And VRC01 class just means these 2 genetic features. We spent 10 years, we've been working on designing such an immunogen since VRC01 was discovered in 2010. And our immunogen that we've designed is a nanoparticle. It's called the EOD-GT8. It's a self-assembling nanoparticle that presents 60 copies of an engineered portion of HIV TP120. And it does sort of meet that we want. And we've done a lot of preclinical experiments mostly in mouse models to find this out, to find that this immunogen has appreciable affinity and avidity for diverse BRCA1 class human naive precursors. Actually, you can take it and sort naive B cells from the blood of humans and pull them out and get the sequences of the antibodies on the surface of the B cells and show, yep, you can fish out B cells that have BRCA1 class precursors on them. That was the other key experiment that we did. We can show that in very difficult mouse models that were sort of engineered to be as much as human-like as possible, this immunogen will prime those responses. It does the job we wanted, and it will induce memory responses that then you can boost further, which is a longer-term aim. So it worked with a lot of different collaborators from lots of different labs. Everyone tried to test it and put it to the screws, basically, and it did work. So we said, okay, we need to find out, will this molecule prime BRCA1 class responses in humans? And this is a very important question because it sets up all that long, complicated strategy that I told you about that many people around the world are pursuing to make an HIV vaccine. No one knows if it's plausible because we don't know if the first shot would work. So, and this molecule, our molecule, was the first time this strategy had been tested. So it's kind of a big deal to get the answer to this question. And so this was the first, we carried out a clinical trial. It was the first in human test of the germline targeting strategy with a self-assembling nanoparticle and a strong adjuvant. Adjuvants are molecules that you add to your vaccine to make them more immunogenic. And we use this adjuvant called ASL1B, which is in other vaccines. It's a very good adjuvant and it's safe. The trial was conducted at the Fred Hutch in Seattle and George Washington University in Washington, DC, from around September 2018 to March 2020. And it's a phase 1 clinical trial. The primary endpoint is assessing adverse events and testing safety. And it was safe and it had a favorable safety profile, so that was fine. But assuming it was safe, the major immunological thing that we wanted to know, the endpoint, was to determine if the vaccine would induce BRCA1 class IgG B cells. IgG is one kind of antibody that B cells have on their surface. In this case, it's the most important one. And that, to figure that out is a very tricky thing to do. You can't do, like for COVID, they do a nasal swab and they can tell if you're positive for COVID. You can't do a nasal swab. You've got to take blood, you've got to do B cell sorting and sequencing to find out what are the sequences of the antibodies on the B cells, and do they have that VH1/2 on the heavy chain, and do they have a 5 amino acid CDRL3? And no one had really ever done that sort of complicated assay as the critical clinical trial before. But we had really great colleagues at the NIH Vaccine Research Center, Adrian McDermott and Rick Kaup, and at the Fred Hutch, Kristen Cohn and Julie McElrath, and they set up this assay and they carried it out for this clinical trial. And so basically this clinical trial is all about them doing this assay to find out what sequences do we have in the vaccine-induced B cells. And the clinical trial had a low dose and a high dose. It had 18 people receiving each vaccine and 6 receiving a placebo, and everyone got 2 shots at week 0 and week 8. And to find out if the vaccine induced BRCA1 class B cells, we took a lot of different samples. So everybody got vaccinated week 0 and week 8, and we took samples at weeks 3 and 4 after the first shot, and at weeks 8, 9, 10, 11, and 16 after the second shot. And we actually took a sample beforehand to understand what the baseline was. And what we're doing with those samples is, all these different samples have B cells in them, and B cells have an antibody on their surface. And our colleagues used single B cell sorting, going through a fluorescence assisted sorter, where we have molecular probes that have fluorescent dyes on them. And single B cells are going through the fluid stream. They're mixed with these fluorescent probes, so you can tell, does this B cell bind to our immunogen or not? And if it does, it goes into the plate. And then we carry out RT to get the sequences of the B cell receptors. When we get the sequences, we did bioinformatic and statistical analysis of those sequences. We made antibodies and did binding assays. Just very briefly, that was sort of the plan for the trial. And it's rather complicated. So just one slide on what they actually were doing. They were taking these B cells and they were labeling them. They were looking, so this would, on this x-axis and y-axis would be the fluorescent signal for binding to our EOD-GT8 molecule, which is the thing that's arrayed on our immunogen. Copies with two different colors to make sure that we have things that were double positive. So they're definitely binding to EOD-GT8, and they're not binding to either one of the fluorescent dyes. And then we also had a mutant of our EOD-GT8 with a mutation in the CD4 binding site such that VRCO1 class antibodies wouldn't bind to it. And so they were gating on B cells that didn't bind to this knockout. And then when they got those, they sorted those single cells into these 96-well plates. And then they carried out reverse transcriptase to generate complementary DNA. And then they carried out stages of PCR to amplify the DNA. And this was a lot of work on their behalf. And they recovered the heavy chain, the kappa chain, and the lambda chain. The light chains, you can either be kappa or lambda. And then we sequenced all of those to find out what the sequencing was. And we paid a company to do that. And then we just have a data set of sequences corresponding to the B cells that were for the CD4 binding site on our immunogen. At that point, until we see the sequences, we can't tell if they're VRCO1 class or not. To tell that, we looked in the sequences to bioinformatic analysis. Do the heavy chains have a VH1/2 with one of these two different alleles? And do they have a 5 amino acid CDRL3? That was the major signal that we were looking for. And so this was the first big readout. If you group, if you just say what fraction of vaccines produced a VRCO1 class B cell versus what fraction of placebo recipients, you can see that in the low dose and the high dose, 18 out of 18 vaccine recipients in the low dose produced a VRCO1 class response, whereas only 1 of 6 placebos. And in the high dose, it was 17 out of 18 versus 1 of 6. And these were highly statistically significant. So this was, when we saw this, we computed it actually. We were looking at the data, and it was because this said, okay, at this level, the strategy is not crazy, and you can turn these things on intentionally. And no one had ever done that before. And then the next thing you want to know is, okay, we turned on the right B cells, but how prevalent are they? How frequent are they in these people's blood? If there's only 1 or 2 of them, it's not really going to work to come in with a boost a different molecule and find 1 or 2 B cells and kick them forward toward BNAB development. You would want a high frequency of them. And what we found is the vaccines did induce a very high frequency. So in the low dose group, you get the first shot. Before the first shot, there really aren't VRCO1 class B cells at all in the memory compartment. There are there in the naive B cell compartment, but there's none in memory. But then 4 weeks after the first shot, all of a sudden you have about 0.01% of your memory B cells in your blood, everywhere in your body, are VRCO1 class. So that's 1 in 10,000 memory B cells is VRCO1 class. And then that goes down a little bit over time from week 4 to 8. You get a second shot, and then that frequency jumps up to about 1 in 1,000. So 0.1%. And this was very consistent across all the different people in the trial. 1 in 1,000 memory B cells in your body would be a VRCO1 cell. And then that would decline a little bit to week 16. And we saw a similar response in the high dose, a little bit more favorable. So I was very excited about this because we don't really know exactly how frequent they need to be for them to make a big enough memory pool that could be then boosted by. But it's hard to imagine that 1 in 1,000 isn't frequent enough. I mean, that's incredibly frequent. Your memory B cells have to be able to deal with all kinds of other pathogens as well. So this is really frequent. So we're excited by that, and we feel like this is very meaningful. I'm going to skip. So then we made antibodies. We made VRC01 class antibodies from the trial and asked how tightly do they bind to our immunogen? Can we show that they're binding, and are they binding tightly? It gives us some information on how well the vaccine is working. And we saw, so these are dissociation constants. That's what's the concentration at which 50% of the antigen is bound. So lower is tighter binding. It's just the way we always think about it. So at week 4 in memory, we made a bunch of VRC01 class antibodies and measured their affinity for EOD-GT8, and the median or the geometric mean affinity is 80 nanomolar. And then at week 8, it's higher affinity. That now you're at a geomean KD of 19 nanomolar. It's tighter binding. Then they get the second shot at week 8, and you can see by week 10 now the affinity has gotten to around 7 nanomolar and 5 nanomolar at week 16. So the second vaccination was causing affinity to increase further. And we know, I don't have time to show you, but separately we could see that the second vaccination was causing the B cells to have more mutations. They were gaining mutation, which is exactly what we wanted to do. So things were sort of working the way we wanted. I'm going to skip that. So what we found was the vaccine has an acceptable safety profile in humans. That was aim number 1. It induced VRC01 class responses in 97% of vaccine recipients. This finding establishes proof of principle for germline targeting priming in humans. And that's very important for making an HIV vaccine, and opens the door for trying it for other pathogens. Like we think it might help to make a universal flu vaccine, for example, or a pan-coronavirus vaccine. We actually didn't expect this molecule to perform we had no idea what it was going to do, but we thought maybe it'll work a little bit, but it won't be a home run. And we'll show that the idea is good, but we need to perfect the molecule and make it work better. And you could still make this work better probably, but it works surprisingly well. And it looks like you could just use it. And that means we could go on and develop the rest of the vaccine sequence. And we feel like at the moment, we don't have to spend more time trying to improve the priming immunogen, which was really great. That's an accelerator for us. Skip that. Yeah, I mentioned that. So, and we recovered the antibodies after the vaccination, and they're helping us to, that information, those antibodies are helping us to figure out what should the second shot be? What should the next immunogen be? I mentioned you have to get a sequence of immunogens to get the mutation to go all the way to BNAB. And so the trial is providing, reagents and information to help guide that. And the last thing is, we do think that this is the first HIV human vaccine trial to confirm its intended mechanistic hypothesis. And we were not trying to make a vaccine. This is not going to protect humans. This is just doing one small step, but it was sort of a big small step. And we think that validates a sort of a reductionist hypothesis or philosophy of trying to take things one step at a time and have a vaccine that has a specific job, and see if it achieves those goals, if it can do the job you want it to do, and then go in again and see if your next shot can do the next job. They need to go that stepwise manner. So I'm going to very quickly tell you what we're doing now, and it's not too long. So that clinical trial establishes proof of principle for germline targeting vaccine priming, the first step. What are the next milestones toward an HIV vaccine? We need to show in humans that if you get a different shot, that you can cause increased maturation in the right direction. I don't know if that'll really work in humans or not. And we're actually doing a clinical right now to test that idea. And then if we show that, the next major milestone is getting to the promised land for at least one kind of BNAB. We need to show that we can induce BNABs in humans for one kind of BNAB, like VRC01 class BNABs. And then ultimately, we need to do that for multiple classes in parallel. So it'll be a rather complicated vaccine. And to do these, to try to make this stuff happen more rapidly, we've started collaborating with Moderna to use mRNA delivered immunogens 4 or 5 years ago, that will help us accelerate our HIV vaccine development, just as the mRNA technology helped the world make help Moderna and Pfizer make mRNA vaccines for COVID so quickly that you've all heard about. And so we have 3 clinical trials launching this year. 2 of them have just launched. One of them, IAVI G002, if we can get that same result that I just showed you with EUD-GT86 but now delivered by RNA. And also in this trial, we're testing the next immunogen, also delivered by RNA, to say, okay, if the priming thing works the way we want, if we come in with a different shot afterward, can you advance the ball further toward BNAP? So it's very exciting. We're also testing RNA priming in Africa, where the vaccine is most, and that's a great collaboration with African colleagues, where they're going to conduct a lot of the analysis to build capacity for cutting edge immunological research. And then finally, we're doing another clinical trial to test the other end of our vaccine strategy, just delivering native trimers by RNA, very much like the COVID vaccine, to find out what do we get from that. It won't induce BNAbs, but we need to understand what we get from that to understand how to use it in these complicated sequential vaccination steps. So I'm going to skip these last 2 and just say that basically what I just said, that we established proof of principle for germline targeting vaccine design. The next major milestones will be to demonstrate sequential boosting and BNAb induction in humans. We do think that ultimately developing a protective vaccine will require multiple iterative human clinical trials, as well as lots of preclinical experiments, obviously, to prepare for those. And that will be very much accelerated by our partnership with Moderna, because they can make a mRNA molecule that is safe to give to a human much more rapidly than one could make a protein that's safe to give a human. And it's also much more economical. And so this will allow us, partnering with Moderna should allow us to do more clinical trials in time, get more feedback from those, and make more rapid progress toward HIV vaccine design. And I'm going to stop there and just say, I've been telling you the story. There's a lot of colleagues behind the scenes and a lot of institutions behind the scenes, a lot of funding and a lot of partners. Thanks to the study volunteers for G001 and the people that did all the 0. Also for G002 and G003 that are happening now and will happen later this year, and for the clinical trial on the trimers. So I will stop there and say thank you. And happy to take questions. Maybe there were, I didn't hear anybody say anything, so I didn't. Sorry about that.
Any questions?
Was that comprehensible? I mean, was that, you guys probably have different backgrounds and maybe some of you are not too immunological. I tried to make it not too specialized, but, was it okay? There's a hand up at least, okay. Raheel.
Thank you very much for your talk. It was really interesting. I think as I'm studying immunology this quarter and doing a lot related to that, it's kind of interesting to see the different aspects of looking from B cells to plasma cells and that whole mechanism. So I just had a question regarding that there. So because you guys are identifying specific B cells that are able to secrete BNAbs, is there a way to, instead of I guess you're targeting those and then trying to get those to try to preserve those in memory. Is there a way to, instead of preserving those in memory, I guess try to keep those and induce some sort of replication at a naive B cell point? If that kind of makes sense.
Well, yeah, it's a good question. I think you're saying, do you have to go through all that? Can't you just get the BNAbs right away, basically? Is there a way to do that? That would be great. So in the case of COVID-19, there's been a lot of there have been similar, I just showed you not germline targeting, but they've analyzed the B cell responses to the mRNA vaccines. There have been a whole bunch of papers that just came out, and they've looked at what the sequences are of the antibodies that were induced, neutralizing antibodies. And many of those they only have a few mutations. They're not very from a naive B cell. They've switched over. So in the way the immune system works, a naive B cell is not going to secrete antibodies, but it's very natural and normal for your body to convert from a naive B cell to a memory B cell to a plasma cell. So that in itself is not a problem. But the problem for HIV is in order to become a BNAB, the antibodies have to gain lots of mutation. In contrast, in the case of COVID-19, one reason the vaccines work so well, and it's relatively easy to make a vaccine, is the B cells can be close to germline. The sequences don't need very many mutations. They just need to convert from a naive B cell to a plasma cell, and on the way gain a few mutations, and boom, they got a neutralizing antibody that takes care of the problem. We need to do that same thing, but we have to gain a lot more mutation before we go to the plasma cell. That's the main challenge. And it's just really sort of it's a structural thing for an antibody to bind to the patch on the HIV spike that doesn't mutate very much. It has to make a lot of mutations to be able to grab just in the right way. You can't, if the antibody touches neighboring parts of the spike that mutate a lot, then it won't be broadly neutralizing anymore. It'll only neutralize things that have, the amino acid that it's touching. And so it just turns out that it requires a lot of mutations for the antibodies to structurally set themselves up to only touch what they should touch, basically.
That makes a lot of sense. Thank you for that response. If no one else has a question, I have one more also, but I think there's one in the chat.
Okay, let's see. Okay, compared to COVID, HIV is spread among a smaller group of people. Differences lead to differences in their viral performance, i.e. evolution, speed, or spike protein expression level. How did you address this indifference in development? Okay. Well, HIV in theory is spread among the same, it can infect, almost any human. The only people that we know that can't get infected with HIV so I mentioned that HIV binds to the CD4 protein and the CCR5 protein. And CCR5, everyone has CD4, and it's a required part of, I don't think there are humans that lack CD4, and you can't make mutations in CD4 to evade HIV infection. But some humans, a small percent, a few percent, have mutations in the CCR5 gene that make HIV not able to infect the cell. So so some people can't be infected by HIV, but everybody else can. So aside from that, though, it's true that it's circulating among a smaller number of people. That's true, but HIV mutates its spike much, much more rapidly. So in one person infected with HIV, there is more genetic diversity by far in the spike gene than there are among all the people on the entire planet infected with COVID right now. I guarantee you that's true. So even though HIV infects fewer people, it mutates its spike so much more aggressively that it's much more difficult to deal with. If HIV would infect everyone around the planet, it would make it even worse. So that would make it much more difficult, but it's already incredibly difficult with millions of people infected. I think that answers your question. But basically, okay, how did you address this difference in developing the vaccine? The fundamental thing that we are trying to induce broadly neutralizing antibodies as opposed to just neutralizing antibodies. The COVID vaccine, they just give one copy of the spike, they get neutralizing antibodies. Even based on the Wuhan strain, they still can get some protection against Omicron. Like if you get 3 shots, you get protection against Omicron. For HIV, that would not work at all. You really have to induce very special broadly neutralizing antibodies. Otherwise, you won't get that cross-reactivity. I'm seeing some head shaking, so I guess that makes sense. Okay. How does HIV develop into AIDS in terms of immunology? Yeah. So yeah, and the person says, I remember it has something to do with the number of T cells. Right. So your immune system, several different arms of your immune system. The two key ones are the arm that makes antibodies and the arm that makes T cells. And both of those, the development of antibodies, the development of B cells, and the development of CD8 killer T cells, which I talked about when they find an infected cell and they drill a hole. Both of those systems rely on CD4 T cells to help them carry out their processes. So HIV, it specifically infects CD4 positive T cells because the primary receptor is on the surface of those T cells. And when it infects those cells, it kills them. It turns them into little virus factories for a moment in time and then just blows them apart and they're dead. And then all the viral progeny go out and find new other CD4 T cells, infects them, kills them, etc. And that it really knocks down your immune system. And you're exactly right. It has to do with the number of, not just the number of T cells, but the number of CD4 T cells. And if your CD4 T cell count gets too low, your antibody part of your immune system doesn't work anymore. And your CD8 your T cell system, that system doesn't work either. And that means you keep off hardly any pathogen. All kinds of pathogens, bacteria, weird yeasts, all kinds of stuff that you normally often just take care of automatically and have no symptoms, they actually could kill you because they can multiply and spread and infect cells all over your body. So people end up dying from weird infections. And when AIDS was first discovered, people came in with weird infections that doctors would never see and they didn't understand. And that helped them realize, oh my God, that person's immune system is not working. That's why they're getting these weird infections. So it's very dangerous because when you're immune compromised, and people are immune compromised for many different reasons, having HIV is sort of like the king of all reasons because it's very, very strong and dangerous. It makes you susceptible to all kinds of infection, and that's how that's why it's so dangerous. Okay, Rohil has another question.
Yeah, I have sort of like I guess a follow-up on that. So HIV kills like the CD4 helper cells. You mentioned that one of the ways to, I guess, prevent HIV from running its course is to bind with those T cells itself and like prevent HIV from binding to them in turn by like, I guess, sending another protein or something to bind to those cells. Does doing that have any sort of effect on the immune system? I'm sure it weakens it to some extent less than HIV, which is why it's possible. But
well, so good question, but you're a little confused, but that's just because there's too much going on. So here's the HIV spike, CD4, and CD4 is on the surface of a human cell. Spike binds to CD4, undergoes conformational changes, and injects genetic material into the cell. Here's the spike, here's CD4. What we're doing is inducing antibodies that bind to the spike in the place where it wants to bind to CD4. So now it can no longer bind to CD4. So we're not binding to CD4, we're binding to the spike and preventing the spike from binding to CD4. So the CD4 T cells they're happy, they're not bothered, they do their own thing normally, and they don't know that anything's wrong. That in this particular case would be the goal. Yeah.
I see, that makes sense. Thank you.
There actually is, there's a guy at Scripps, Florida, who's working on not a vaccine, but a treatment for HIV, which is based on giving them CD4. And he made an engineered version of soluble CD4. So if you have a spike, the engineered version of CD4 would just bind to the spike and prevent it from binding to CD4 positive T cells. And that kind of therapy is interesting, but it does have some potential risks, which if I give you a shot that contains human CD4, you might respond by making antibodies, the shot I just gave you, and they would be autoantibodies. And very dangerous, you could all of a sudden have autoimmunity. And so I'm concerned about that strategy for that reason. In animal experiments, they don't see evidence for induction of autoimmunity, but they can't do animal experiments in hundreds of thousands of animals. And if it only happens in a small fraction of the people that you would immunize, that you would deliver this CD4 to, you still wouldn't want to do it because you could actually cause that person a lot of harm. So that's sort of the nature of your question. It's just that when you do a real, an actual vaccine, we don't have that problem. Okay, there's one other, I think you guys have another session, but there's one other, why did you choose to target the VRC01 region over any other low mutability regions? Great question. We're actually focused on 4 different ones. We have 4 different projects going full steam, trying to do the same thing so we can induce multiple classes of different VNAVs. And the VRC01 project just happened to start first. So. All right, well, thanks you guys. I don't want to stop you from going to your next session, and I'll try to attend I think there's something at noon, right? For networking. I'll attend that in case anybody wants to talk more.
Okay, thank you so much, Dr. William Shi. And yes, we have the next session and the PhD panel happening now, and I dropped the link in the chat. So thank you for everyone for coming.
Thanks everybody. Cheers.